such as it is.
Thank you. Thank you for inviting me. And let me know if you can't hear me or if you want me to speak up.
You're fine.
Okay, it's all yours.
All right. Oh, there it is. Okay, so I'll tell you a little bit about the research I've been doing on Noctuid moths as a part of my PhD, which I just finished. And some of you may actually recognize this photo. This is Anza Borrego Desert State Park and the Steel Burnan Desert Research Station down in the bottom right-hand corner, where I've done a lot of the field work for my PhD.
We are not seeing it as a slideshow.
Oh, okay. Let me
share
try
again.
that again.
I think what you have to do is, yeah, you have to share from the slideshow.
How's that?
Yes.
Okay. So now you can see the first slide with Anza Borrego and the Steel Burnan station down there in the bottom right-hand corner. Did a lot of my field work there. And I didn't intend on working on the entirety of Noctuidae because that would just be foolish for someone to try to work on 12,000 species as a part of their PhD. I really just wanted to work on this part of Noctuidae, the Styrianae, which is one little tribe in it, or it was a tribe in it before I messed it up. And these are really gorgeous moths, gorgeous adults with these golds and oranges and purples. And more than that, they're all basically in the deserts of North America. And I love the deserts of North America. So I figured I would study this group and it would give me an excuse to get out into the desert. And if you're not intrigued enough just by the way that these adults look, they are also the subject of Charlie Hoag's PhD dissertation. Some of you may have known Charlie Hoag. He was the senior curator at the LACM for a while and Jim's father. And in addition to that, this group is just fantastic because the caterpillars are these extreme diet specialists, and they are typically feeding on a single species of host plant, and they're all mostly feeding on the flowers of their host plants. And all of their host plants are in the same family, the sunflower family or the Asteraceae. And some of them, like the caterpillar in the top right-hand corner in the genus Styria, are just these fantastic mimics of the flowers that they feed on, like the Styria in the bottom left-hand corner. You can see with just perfect sculpturing, perfect texture and color to match the flower that it's feeding on. And it's even matching it behaviorally by curling up in the flower head that it's partially eaten. And again, you can see the Styria caterpillar here in the right-hand corner, bottom right-hand corner, curled up on the flower head, just perfectly matching the flower and maybe getting away from a bird that's looking for a meal. So I figured this would be a really fun group to study. I could study plants, which I'm quite fond of, and get back out into the desert. And in addition to that, I thought it would be kind of cool to study how their diet breadth has changed over evolutionary time, to see if maybe they're fated to feed on a single plant species, or if over evolutionary time, they're able to kind of branch out and feed on a different plant species and increase their diet breadth. But in order to do that, I needed a phylogeny for the group, and there was no phylogeny for the group. So I had to get a phylogeny for the group first. And these phylogenies are, they're really useful for answering a lot of these academic questions, but more so than that, they're really useful for just understanding the world that we live in. So these phylogenies are really like biological infrastructure that all of these other ecological and evolutionary studies are based off of. And if you have this phylogeny in hand, you can answer and ask all these ecological and evolutionary questions, like the one that I was interested in looking at the diet breadth of these species over time. And besides these academic sort of benefits of having a phylogeny, there are also just these really practical benefits if you have your taxonomy based on phylogeny, if you have your taxonomy grounded in evolutionary history. So let's say you have two species in the same genus. If they're classified in the same genus and you know what one of them feeds on as a caterpillar, as long as that taxonomy is based in evolutionary history, then you can make a pretty good guess as to what another species classified with it feeds on. Because if they're classified together, they share a lot of evolutionary history together, and thus they probably share a lot of the same behaviors and ecological interactions. So you can make these predictions about what things feed on and better conserve species for which you don't know the life histories of. And in addition to that, let's say you're working for the USDA as maybe inspector at a port somewhere, and this weird ship comes in with a weird moth on it. If you know what genus that moth is in, and you know what other species in that genus do, you can make a pretty good prediction as to whether or not you should be worried about that species becoming a pest or becoming invasive in the country. So some practical benefits of having phylogenies in that you can have your taxonomy based off of that phylogeny. So I needed to get a phylogeny for this group to study these questions that I was interested in, but more so than just the phylogeny of this group, I needed to get a phylogeny of the encompassing group, the subfamily Amphipyrenae. So I set out to do that, and that's kind of where everything went wrong with my PhD. So I'll tell you about my PhD in 3 parts, a short little jaunt, which I thought was going to be a short jaunt in Amphipyrenae, and how that kind of led us down this detour across the whole family Noctuidae. And then once I had wasted 3 or 4 years of my PhD on that, I was able to get back on the Stereines and study that group with those wonderful flower feeding caterpillars that I was so enamored with at the beginning of my PhD. So first, the Amphipyrenae. This is a pretty diverse group of moths, and they're actually a little too diverse. Systematists have known for a long time that they're probably just this junk drawer taxon or this trash bin taxon, dumpster taxon, waste bin taxon, whatever you want to call it. Basically, it's this group of species that are classified together, but people have known for decades, for centuries, that they really have no business being classified together because they're not actually closely related to each other. So this taxonomy of this group is probably not reflective of the evolutionary history. And you probably have a junk drawer in your house somewhere with all these things that you just can't figure out another place for in your house, and you just put them in this drawer together, but there's no relation amongst all the things in it. It's the same thing with this subfamily Amphipyrenae. So I wanted to infer molecular phylogeny for this group because the morphology of the group is just so confusing. It's, I'm not insulting the taxonomists or systematists that have worked on this group before. It's just a really difficult problem to understand all the diversity of this family Noctuidae, all the morphological diversity, and properly classify everything. But with molecular data, you can kind of cut through that confusing morphology and make sense of it. So I figured I would infer a molecular phylogeny for the group, get that taxonomy reflective of evolutionary history, and then I could work on those Stereines and look at how their diet breadth has evolved over time. So I just went through the checklist of the moths of North America and tried to get as many genera as I could to get a good sampling of the diversity of the subfamily. But more so than that, I tried to get these higher level taxa, and I'm sure some of you're all familiar with Linnaean taxonomy, with these different categories of life, with life at the top being the most inclusive group, and as you go down towards the bottom with species, that's the most exclusive group in this list. And in these really hyper diverse groups like insects and moths and butterflies, we take advantage of these other groupings just to kind of break them up into smaller, more digestible chunks. So I wanted to get representatives of all these tribes and subtribes within Amphipyrenae, all these higher level taxa, and they all have their own special suffixes. So our subfamily Amphipyrenae here has 3 tribes, Amphipyrenae, Staphydini, and Stereinae, and then a few subtribes within those latter 2 tribes. So I wanted to get not just all the genera that I could, but representatives from all these bigger chunks of diversity, these higher level taxa, the tribes and subtribes. So what we ended up getting for taxa in our analysis was we had 120 Noctuids, more so than just Amphipyrenae. We had representatives of many other subfamilies within the family, just in case some Amphipyrines didn't truly belong in Amphipyrenae. We could see which subfamily they truly belonged in. And we ended up having 57 of the 76 Amphipyrine genera. We were able to add our data to a larger data set of Noctuidae that previous researchers had built. And 55 of those Amphipyrine genera were new and had never been included in a molecular phylogenetic analysis before. And importantly, 47 of our Amphipyrine genera were represented by their type species. So the type species is the anchor of a genus. Wherever that type species ends up in your analysis, wherever it ends up in the tree of life, that's where the genus ends up. So we had a lot of taxonomic weight for this study. We could reassign genera based on where those type species ended up in the tree. And importantly, we had representatives of all the tribes and subtribes within the subfamily Amphipyrenae. And we just use a standard gene set that has been used for over a decade, almost 2 decades now, to build phylogenies of moths and butterflies, and did very standard software analyses to build the phylogeny, so nothing fancy or new there. And in addition to that, we basically tried to use adult and larval morphology and life history data, ecological data, to either support or refute what we found with our molecular data, because we thought that we might have some odd results, and we did have some really odd results. So the tree that we were able to build is shown here, and everything that's red in the tree, those are lineages that were classified as Amphipyrenae before our study. And everything that's black, those are lineages that were not classified as Amphipyrenae. So if Amphipyrenae were just a good group of species that were most closely related to each other, you would see one chunk of red in the tree. But instead, there's red all over the tree, all over the family Noctuidae. So Amphipyrenae is just this wildly unnatural grouping with representatives of 10 different subfamilies within it. And this is just, I mean, this was such a mess, far more of a mess than any of us, myself or my co-authors could have imagined. We had one of the curators at the Canadian National Collection on our paper, and it just totally exploded his idea of Amphipyrenae systematics, but also systematics for the entire family Noctuidae. So we had some work to do if we were gonna make Amphipyrenae a monophyletic group, just this good group of species that are most closely related to each other, and also to fix the taxonomy of all these other subfamilies in Noctuidae that turned out to not consist of all of their rightful members. So the tree that you're seeing here on the left, that's the same color scheme that we saw before. Everything that's red, those are things that were classified as Amphipyrenae, and everything that's black, those are things that were not classified as Amphipyrenae. And then in the tree panel on the right-hand side, you see different clades colored and their labels are bolded if they received Amphipyrine genera. And then you see images of the moths for those different groups that received Amphipyrine genera. And all of those moths used to be in the same subfamily in Amphipyrenae. So Amphipyrenae is at the bottom of the tree there, and that's less than half of its diversity. We ended up removing over half of the diversity of Amphipyrenae and reassigning it to other subfamilies across the family Noctuidae. Most of it ended up in Styrianae down in the middle of your screen there, and that's the group with those wonderful flower feeding caterpillars. And then there were a few other genera that got placed in other subfamilies like Nacopa here. We just reassigned it to Noctuinae. And then Acopa, we reassigned it to Bryophyllinae, and so on. And we had a lot of really interesting things that we found in this study, but just 2 in particular that I'll mention. So we had this group of 4 genera that were formerly classified as Amphipyrenae, grouping in the subfamily Acontiinae, and we described a new tribe for them. All of the caterpillars of this group are specialists, diet specialists. They feed on the flowers and seeds of species of Malvaceae, which is the hibiscus or cotton family. They're all residents of the deserts of Arizona, New Mexico, and Mexico. They don't quite make it into California. But with our molecular perspective, we're able to kind of make sense of why this group is special. So they have a different number of prolegs than Acontiines, even though they grouped with them. They have 4 of these fleshy pairs of prolegs here, 1, 2, 3, 4. Acontiines usually have 2. So they grouped with them molecularly, but they're quite distinct based on their morphology and their life history. So we described this new tribe for them, Camicleinae. And in addition to that, we found this single genus Cropia, which grouped towards the top of the tree here outside of any known Noctuid subfamily, which was quite a surprise. But when we dug into the literature, it actually made a lot of sense that Cropia didn't group with any other subfamily. Cropia has actually long been known to be kind of a weirdo in Noctuidae. The male genitalia are kind of bizarre, and the caterpillar has this bizarre combination of characteristics with a humped segment when in yellow towards the rear of the caterpillar, and a really constricted prothoracic segment here. And it has this kind of odd threat response where it rears up with its thoracic legs and basically just vomits out some of its pre-digested food in its crop. So this really weird combination of characteristics for Noctuidae. And we thought about describing a new subfamily for it, but we didn't have really strong molecular support for where it belonged in Noctuidae, and we didn't have the type species for it. So we didn't want to describe a subfamily called Cropiinae and then turn out to not have the genus Cropia belong in it. So we decided to wait until we could get stronger molecular support for its placement, but also importantly, to wait until we could get the type species of Cropia included in our analysis. So in the end, we ended up reassigning 31 genera to other areas in Noctuidae based on molecules and supporting those moves with morphology and life history data. We ended up newly recognizing 2 subfamilies, the Sterinae, those wonderful flower feeders, and Grotalinae, described 1 new tribe, Camiclini, and we identified 1 potentially undescribed subfamily with Cropia. And this has since been published, and I really can't stress how wild it was that this single subfamily had representatives of 10 different subfamilies within it. I thought I just contaminated all my samples and it ruined my career as a systematist, but it all worked out looking at the morphology and life history data. And we were just so intrigued by this, we had to dig deeper and try to build a phylogeny for all of Noctuidae, try to get more genera from around the world included in our analysis. And that's what we did for the second part of my PhD. We just dug down into Noctuidae as far as we could and ended up building the largest phylogeny of Noctuidae so far. So that picture I showed you before of the Amphipyrenae diversity is actually a pretty good representation of the diversity of moths in the entire family Noctuidae, with representatives of 10 different subfamilies in it. The group is extremely diverse. The caterpillars too feed on all sorts of different parts of plants and all sorts of different types of plants. There are over 12,000 species of noctuids in the world. And in addition to being extremely diverse, they're also extremely abundant in terrestrial ecosystems, being major food sources for birds. They're so abundant that they're actually a critical food source for grizzly bears in Western North America and for Aboriginal humans in Australia with the bogong moth there shown on the right. In addition to that, they also eat a lot of our food. Some of the world's worst agricultural pests are in this group that cause billions of dollars in crop losses around the world, like the corn earworm on the top and the fall armyworm on the bottom. And you can see the adults of those 2 species there nectaring. And it turns out that noctuids are actually really important pollinators at night. They're actually the most important nocturnal pollinators in some landscapes, like agricultural landscapes. And despite being so diverse, so abundant, being so important in terms of crop pests and being so important in terms of ecosystem services with pollination, there still isn't a good phylogeny for the group. The best was ours, and ours was awful. We only had 10% of the world's genera included, which is a resounding F, not even close to a D, and that could not stand. We had to get more genera. We tried to sequence more genera, tried to get DNA for more genera by doing our own sequencing, but also mining all the publicly available DNA that we could find on GenBank and the Barcodes of Life database. And we tried to increase our geographic sampling because our previous study was pretty much just focused on the Nearctic or the North American fauna. And we tried to increase our higher level taxon sampling, tried to get all the subfamilies, tribes, and subtribes included in our analysis. And I was quite keen on looking into more Camiclini genera, trying to find more of them around the world, and trying to get that type species of Cropia to see if it truly did represent a new subfamily. And I was really interested in looking around in other little junk drawers across Noctuidae. And critically, we needed more experts on this paper because we could hardly handle Amphipyrenae. And if we were going to do an analysis of the whole family, we needed to get noctuidologists from around the world that actually had expertise in the family. So the tree that we inferred is shown here, and all the red lineages this time are newly added lineages to our previous paper. So everything that's black, those are lineages that we had included in the Amphipyrenae paper, and everything that's red are things that we were able to add to that data set. We ended up having 463 total taxa in our analysis, with 341 noctuid genera, which is just about 1/3 of the world's noctuid genera included. And most importantly, we had 70 of the 76 higher level taxa in Noctuidae. So 20 of the 21 subfamilies, 32 of the 35 tribes, and 18 of the 20 subtribes. So excellent sampling of these bigger chunks of diversity across the family. And we found a lot of interesting stuff that I can't go into detail completely. We also did a great job of increasing our geographic sampling. In our previous study, it was mostly Nearctic, but we did a great job of getting into areas like the Neotropics, the Afrotropics, and Australia, which were particularly poorly sampled in our previous study. So we had a bunch of interesting things that we found, but I'll just talk about a few of them here. So we found 7 new Chamaecleon genera from around the world in the Neotropics, the Palearctic, and Australia. So before, this was just a group of 4 genera that were all in North America, but now we have these new genera from the Neotropics, Palearctic, and Australia, so increasing our biogeographic understanding of this group. And one really interesting thing was that we had this single species, Azenia virida, grouping here with Azenia in quotation marks. So Azenia virida grouped apart from all the other species in its genus. All the other species of Azenia grouped in a different subfamily, and that's a big taxonomic no-no. You want all of the species of a genus to belong in the same subfamily. But this and 4 other genera in our analysis actually had species that grouped in different subfamilies in different parts of the Noctuidae tree. And this is quite surprising. So we figured we have to figure out if we can support this molecular finding with life history data or morphological data. And we knew that all the Azenia species that we had life history data for, they all feed on dodder or cuscuta, which is this little orange strand shown here. It's this parasitic plant that you may have seen kind of growing over other plants looking like spaghetti. So all the caterpillars of Azenia look like that, but we don't know, or we didn't know, what the caterpillar of Azenia virida looked like. And we figured if it's a Chamaecleon, it's got to be a specialist on Malvaceae, just like all the other species of Chamaeclini. So we went digging around for hosts, and it turned out that there was a single photograph like this one that existed of the adult of Azenia virida sitting on a globe mallow or spheralcia out in Anza-Borrego in the book shown here. There was just a single photograph of the adult sitting on that plant. And so we figured it looks a lot like the plant, and it's a malvaceous plant. So that's probably a pretty good guess as for the host of Azenia virida. So I went out to the Chuckwalla Mountains when the globe mallow was in bloom, and I went looking around for caterpillar damage on the flower buds and opened up hundreds of these flower buds and wasn't finding anything, and just kind of felt sorry for myself and just sat down in front of a globe mallow and just stared at it. And then I saw this little wasp come by. And as you know, wasps love to lay their eggs in caterpillars. And this wasp landed on a flower bud and plunged its abdomen into the flower bud. And then when it flew off, I ran over to the flower bud and opened it up. And inside, there's a little caterpillar, this little grub-like caterpillar. And I brought it back to the lab in Connecticut, and I grew it up into a big, strong caterpillar. And it looked just like the other Chamaecleons, this kind of hairless, plump caterpillar with 4 pairs of prolegs and a nicely developed anal plate towards the bottom there, just like the other Chamaecleons. And it's a specialist on Malvaceae. And we got DNA from the caterpillar, threw it into our analysis, and it was a perfect match for the DNA that we had from the adult in our analysis. So great support for moving Azenia virida into the Chamaeclini, and great support for it having nothing to do with the other Azenia species, and a great example of how phylogenies can be used to predict unknown life histories. We knew this thing was grouping with Chamaecleons that all eat malvaceous plants, and so we just predicted has to eat Malvaceae and maybe globe mallow, and it turned out to be correct. In addition to that, we found this group of genera in the Copobatha clade that we called it. They were formerly classified in Eustrodionae down here, but they didn't group with them. And this was quite odd, but made complete sense when we looked at their morphology. So we ended up describing a new subfamily for them, Copobathinae. And it turns out the caterpillars are all specialists on Acanthaceae, unlike any other Eustrodine, and they have a different number of prolegs than the other Eustrodines. So we ended up describing a new subfamily for them based on those and other characteristics. And this completely altered our understanding of Eustrodinae. Before our analysis, you would have thought of Eustrodines as having radiated in the Nearctic or Neotropics, and basically all being specialists on Acanthaceae, which is the mangrove family. But after our analysis, with these Copobathinae genera removed, turns out Eustrodioines are basically only a temperate taxon with species that feed on grasses and sedges as caterpillars. So completely altered our sort of understanding of the biogeographic hotspots of these groups and their ecologies. And you can actually find this caterpillar, Copobatha lixiva, and its adult in the LA basin. There's a lot of hidden diversity, even in the metropolis of the LA basin. And another really interesting thing that we found was we were able to include that type species of Cropia, and it grouped with the other Cropia from our previous analysis. So once we saw that, we started digging around in the morphology of these species, trying to find something that we could use to describe it, to describe this new subfamily, but also to differentiate it from all the other subfamilies. And what we found had to do with these courtship brushes. So you can see the brush here in the image on the top left. Some males of Noctuid species have these brushes that'll unfurl right before they're about to mate. And it's basically just a fail-safe method to make sure that to tell the female that they're the same species, and basically so they don't make a mistake and waste their gametes on the wrong species. And these brushes are typically found in species that exist in areas where there are a lot of other closely related species, and they're basically an adaptation to prevent those mating mistakes. And you can see the ultrastructure of the scales that are a part of those brushes. It's basically just these big wicks that evaporate these pheromones. So we looked at the courtship brushes in Cropiinae, and we found some interesting things. All the courtship brushes in Noctuidae before our study were thought of to look like this. So there's a brush that you can see here, which is attached to a lever, which is attached to the posterior lateral corner of the second abdominal segment. So we're looking up from the bottom of the moth, the abdominal segment 2 here, third abdominal segment, and fourth abdominal segment. And when we looked at Cropia, it was totally different. So there's a brush, just like all the other Noctuid brush organs, but there are 2 levers instead of 1 that have a junction in the posterior corner, but they don't actually attach to the abdomen until the anterior portion of the second abdominal segment. So 2 levers instead of 1, and a different attachment point. And this is a totally different configuration of this courtship brush and lever system, totally different than any other Noctuid. And we never even would have thought to have looked at this if it weren't for this molecular perspective. And we had a co-author on this study from the British Natural History Museum, and he's been working on these brushes and levers for almost 30 years, and he'd never seen anything like this. So ended up describing the subfamily Cropiinae and using this unique lever system and brush system as a means for differentiating it from all the other subfamilies in Noctuidae. In the end, we reassigned 36 genera, about 10% of all the genera in our analysis, to other groups within Noctuidae. We described 2 new subfamilies, found 7 new genera in the Camiclini, expanding it out of the Nearctic, and found a bunch of other groups that are perhaps worthy of having new tribes and subtribes described for them, but for which we needed more data to do that. And this has since been published, and just want to stress how much of a team effort this was, not just of myself, but all my co-authors and everybody that sent us specimens. Dave Weichel included has sent us specimens, and just want to thank him and everybody else that really made this study possible. And once that was done, and I had chewed through 3 years of my PhD, I was able to get back to the Sterioines finally, which has those wonderful flower-feeding caterpillars. This group has been studied a couple times in the past. I mentioned once by Charlie Hogue, and that's the most comprehensive treatment. And it was studied again about 30 years later by Robert Poole. And being about 30 years after 1995, I figured it's about time to study it again. And both of these guys had come up with phylogenies for the group based on morphology. But they both noted that their findings were preliminary and that the morphology was just kind of confusing to figure out, and that a lot of their relationships in their phylogeny were kind of unstable. So they made a lot of changes of their own to the taxonomy of the group, but they also basically pushed a lot of them for future systematists to tackle once they could get more data. So I figured, just like with Amphipyrenae and Noctuidae, I could leverage this molecular data to build a phylogeny for the group. And in addition to that, I figured we could increase our knowledge of just where these species are in the world. So if you look at the maps in Poole's work, you'll see a lot of maps of the species like this, where there are specimens that go up to the US-Mexico border and then nothing until Mexico City, or they go up to the US-Mexico border and then there's just nothing. So maybe the border is this fantastic biogeographic barrier, or maybe people just haven't sampled there. And I figured it was the latter, and we could probably pretty easily fill in that knowledge gap. So for this part of my dissertation, just wanted to increase our knowledge of where these species are in the world and infer molecular phylogeny for the group, but also wanted to include a lot of this new life history information that we'd uncovered over the past years, searching for caterpillars out in the desert. So I figured I would go to the museums to try to get representatives of all the Mexican species. But I went to the San Diego Natural History Museum, the LA County Museum, the Smithsonian, the American Museum of Natural History in New York, the Canadian National Collection, and the National Collection of Insects in Mexico City, and all their specimens were too old for me to sample. So I figured I'd have to go to Mexico to actually get fresh specimens to include in our analysis. But the collection data on the labels of these were a little too vague for me to base a field collecting trip off of. So I figured I had to get more fine detailed locality information. So I went to iNaturalist and just looked at all the noctuids in Mexico that flew about the time that the sterines flew, and ended up making a few hundred identifications of sterines and getting a really good idea for where I should go. Also made a few friends on iNaturalist that helped me out in Mexico, that took me out into the field or provided me with specimens. And I was able to go down there and do some collecting, did some collecting of the adults during the day because they just like to sit on their host plants, basically can't be bothered to fly away at all. But mostly I just did collecting at night. So I drove around in my little car, my little rental car with my 2 light traps made by Norris Bloomfield. Some of you may know, collector out of San Diego. And I would just drive for 8 hours a day so that I had enough time to charge the batteries from the cigarette lighter in the car. And I would set them out, also driving that long so I could cover as much geographic area and get as many species as I could. And then I would set the traps out. And then while those were out, I would go around to gas stations and convenience store lights trying to supplement whatever I got in my traps. And I was able to do some collecting in basically over 1/3 of the length of Mexico. One of my friends that I made on iNaturalist is shown there in Toluca. That's Juan Carlos Garcia Morales. He took me out to one of his favorite collecting spots. Also did some collecting in San Luis Potosí, in Coahuila, and Guanajuato, which turned out to be the hotspot of sterine diversity. And I ended up getting a lot of the Mexican endemic species to include in our analysis, and also specimens from Mexico of species that also occur in the United States. So good sampling of the Mexican diversity of this group. And in the end, we were able to sample 10 of the 14 genera with 65 of the about 100 or so aster feeding sterines. And we were able to include 12 of the Mexican endemic species. And we used a very similar gene set and set of analyses as our previous studies. So nothing new there. And what we found 3 main groupings in Sterinae. We found this single genus, Numigenia, which is out in Arizona and Mexico, outside of all the other species in Sterinae. And then there are these 2 bigger groups of genera, Plagiomimicus and allies, and Styria and allies. And this arrangement of genera made total sense with the life history information that we'd been able to figure out. So Numigenia is the only genus in this group that has species that only feed on leaves, that don't feed on the flowers or fruits of their host plants, whereas all the members of Plagiomimicus and allies and Styria and allies are all feeding on the flowers and or the seeds of their host plants. So that arrangement made sense. What didn't make sense was what we found in Plagiomimicus and allies, which turned out to be this other little junk drawer that I was not expecting. So what we found was inside that junk drawer, we found this single genus Lineostriae styria on the outside of everything else. And that made total sense. Poole, in his work, Robert Poole, had separated that from Plagiomimicus, and Hoag had actually hinted at that Lineostriae styria should be separated from Plagiomimicus. So the fact that that was on the outside of everything else made total sense. But inside Plagiomimicus, it was a little more confusing. We found Plagiomimicus coming out in 4 different areas in this group. And inside of it, we had Chrysoestia, this other genus, coming out in 3 different groups. And then nestled within one little chunk of Chrysoestia, we found the single genus Xanthothrix, which turns out to be day-flying, the only day-flying group within Sterinae, and a California endemic. So this was a little odd, but we just went through the life history data that we had and the morphological data that we had, and we started to make sense of it all. So looking at Plagiomimicus, the group of Plagiomimicus up towards the top, and comparing them to the Plagiomimicus on the bottom, all the Plagiomimicus in that top group are external feeders, and they're all feeding on asters in the tribe Eupatoriae, which is totally different than the Plagiomimicus on the bottom, which are all internal feeders, and they all feed on asters in the tribe Helianthiae. So that separation between those two groups of Plagiomimicus made total sense. And thankfully, someone had described that group of Plagiomimicus in a different genus in Polenta. So we just resurrected that genus, didn't have to describe a new genus. And then just going down here, looking at Chrysoecia and Xanthothrix, turns out the caterpillars are nearly identical to each other. So figured it makes no sense to have a distinct genus for Xanthothrix, and we just synonymized it with Chrysoecia based on the larval morphology. And then moving down to this other group of Chrysoecia with this totally bizarre caterpillar that we were able to find in the last couple of years, looks nothing like the other Chrysoecia and looks nothing like any other Noctuid caterpillar that I've ever seen or that my advisor Dave Wagner has ever seen, and he wrote the book on caterpillars. It really looks like a leaf beetle larva, nothing like the larva of a moth or a butterfly. And you can see the caterpillar in the photograph on the bottom, feeding head down in the seed head of a cosmos, and with its butt up in the air, and this little pointy bit almost mimicking the points on the awns surrounding the seed head. So good flower mimic, and separating it from the rest of Chrysoecia based on its totally bizarre morphology. And again, someone had described these in a different genus beforehand, so we just resurrected that, saved us a lot of time. And we didn't quite know what to do with these other two groups of Plagiomimicus. There's still a lot of work to do with this genus. It's still an unnatural grouping. We didn't have strong molecular support for the placement of those two chunks of Plagiomimicus, and we don't know what the caterpillars feed on, so we couldn't make sense of it based on larval morphology or life history. So some future systematists will have to figure that out. And over the course of this, we actually found a new species of Steria. Those are those wonderful flower mimics that I mentioned before. And there's a species of Steria, Steria diari, that you can find in Southern California, shown here. And the caterpillar is not just a mimic of the plant at one stage, it's a mimic of the plant throughout its development. So when the flower is in bloom, the caterpillar will match the flower. And when the plant is in seed, the caterpillar will match the plant in seed, changing its color to this kind of reddish brown color and changing its behavior. And you can see the caterpillar in the image on the right, curled up on the stem there, doing its best job mimicking a seed head. So Steria diari is found in California, but it's also found in Arizona, New Mexico, and West Texas. And my advisor, Dave Wagner, and my lab mate, Tanner Matson, were out in West Texas, and they found this caterpillar that kind of looked like Steria diari, but a little bit different. And it was feeding on a different host plant. So they collected it, and they collected adults of it, and I got some DNA out of them, included them in our analysis, and they turned out to be distinct from the other Steria diari in our analysis. And we ended up describing a new species for it, which we called Steria brava, after the Rio Bravo, which is the Mexican name for the Rio Grande, and what we think is kind of the center of Steria brava's geographic range. And we found a bunch of different good characteristics to use to separate them. So if you look at the thorax of Steria brava, it's got some nice white frosting to the scales, more so than Steria diari. And if you look at the hindwing, there's far more brown in the hindwing than there is in Steria diari. And Steria brava turns out to be the smallest of all the Steria species. So I ended up describing a new species for it. And probably the coolest thing that I did as part of my PhD was I was able to include my mom. My mom taught painting and drawing for 40 years at the Massachusetts College of Art, and she did the illustrations of the genitalia of our new species, with the female genitalia shown there in A, and then the different parts of the male genitalia shown in B, C, and D. And she didn't just draw them in any old style. She drew them in the style that Charlie Hogue had drawn them in his work, which is really just a meticulous and fantastic study of the Sterines. So an homage to Charlie Hogue and his fantastic work. So in summary, we were able to build the first molecular phylogeny for this group with 80 of the 139 described species, with a lot of the aster feeding species included. We reinstated 3 genera, noting 1 genus that needs to be described. We revealed 15 new life histories, revealing what the caterpillars feed on, And we described one new species and noting 4 others that probably need to be described. So in the end, after all of that, there's still a lot of work to do on aster feeding stereines. I would love to continue studying them, especially in Mexico, where they're probably most diverse. That's where asters are most diverse. And I'm working on getting a couple species level phylogenies along with their natural history of a couple other subfamilies out. We have a lot of data on a couple other subfamilies of Noctuidae. And I would love to combine our Noctuid phylogeny with all of these species. With the Noctuid phylogeny, it's going to be coming out from a group at University of Florida. The one, well, not the one, but a big limitation of our phylogeny is that the backbone is really weak. We don't have strong support for it, so we don't know the relationships among the subfamilies. But this group out of University of Florida has a really solid backbone. So they know all the relationships among the subfamilies. So if we can just add all the species that we have in our data set to their data set, that we can get this really solid phylogeny of thousands of species that will just be this just immense piece of biological infrastructure that you can use to answer all these cool ecological and evolutionary questions. And of course, I would just love to continue expanding our phylogeny of Noctuidae over the course of this work. We just found all these weird genera from around the world of Noctuidae that we just didn't know where they would fall out in the tree, but we had some guesses. So it'd be really fun to just continue adding more genera from around the world to our data set. And with that, just want to thank everyone that has helped me along the way and everybody that has funded me along the way. And thank you for inviting me here to talk today. And I'll take any questions if you have them.
Maybe we want to stop share. There you go.
Did you see much evidence of hybridization between closely related species?
That's a very good question, and a question I would love to know the answer to. Every once in a while, I see a moth that looks like it's kind of intermediate, especially in Styria, which seems like it kind of radiated pretty recently and pretty quickly. So my guess is there's probably a fair amount of hybridization in Styria in that genus, but I'm not sure.
Yes, would they feed on any, like the ones that feed on the Malvaceae or Asteraceae? Would they feed on any cultivated plants in that family?
Yeah, there's a Plagimimicus spumosum, I think, that feeds on sunflowers. So, you know, Helianthus annuus.
Kevin, that was fantastic. Thank you so much. I wondered, how are you fixed for continued funding for your project?
Terribly. I'm always looking for money.
I bet. Yeah. Well, best of luck on that.
Thanks.
Okay, I apologize. I had to step away there for a minute. We would like to say thank you very much for a very interesting and educational presentation. Most of us, that's not right. A lot of us find this type of research a little bit over our heads. As I had mentioned, we have a lot of rough amateurs, etc. You did a great job in explaining it. Okay, hopefully I'll remember some of it. And do we have anybody else that has any comments or questions for Dr. Keegan?
I have a comment, and I realized most of us couldn't do this kind of research ourselves at all, but this seems like really terrific systematic research, which is changing our view of all kinds of things in our favorite insect groups. And I think one of the interesting and really fun things about his presentation was the credit to people who have come before him. Charlie Hogue, we talked about in that respect, but on one of his early slides, I noticed Noctua pronuba, described by Linnaeus himself. And we recall, maybe 20 years ago, when this European moth that had been introduced to the United States a long time ago finally arrived in California. And this was quite exciting finding this because it's a pretty flashy moth.
Yeah, yeah.
And then a few years later, we had the great privilege to go to the Linnaean Society in London, where they have Linnaeus's collection in a vault down the basement. The Swedes are quite unhappy about this, that the Brits bought it from his estate, but there it is. And as Sandy's pulling drawers open to see what's here and there, lo and behold, there's a Noctua pronuba, a drawer of them in Linnaeus's collection. And so that if we're talking about Noctuids, that's a really good place to base it.
Yeah.
I might want to add to this, in your survey of museum collections to survey, I'm a volunteer at the Santa Barbara Museum of Natural History. And we have a vastly growing collection of Lepidoptera, and we have already contributed all of our butterflies to that. And I'm in the process of tagging and organizing the moths to bring over also. So in a couple of years, this will be a very good regional resource. But we also have collectors, Tom Dimmick, Bob Filer, and we've collected a little bit in Mexico. But you will find all kinds of goodies in there going back quite a bit of history.
Yeah, I've been meaning to get over to the Santa Barbara Museum.
Well, if you come this way, give us a call. Dave Wagner's stayed here, so you should too.
We've got a guest room.
We
You're
have
welcome.
it is a
And a guided
Lepidoptera
tour.
guest room.
Oh, thank you.
I've seen their moth collection, and it's gonna take a couple years just to move it to Santa Barbara.
Yes.
Yes,
I know.
that's right. There
I'm in
are
about my third year.
there are tens of thousands from 30 years of blacklighting in Malibu and Santa Barbara.
You would enjoy that very much. And they have a nice pool besides the view. Okay, any more questions?
I have a comment. I'm very glad to see your use of iNaturalist because I'm a strong contributor to iNaturalist, and it's nice to see that it's actually used on the other end. I just put things in.
Jim?
Yeah, I couldn't have done that study without it. I would not have known where to go.
You mentioned encountering potentially useful specimens that were too old to gather genetic information from. Could you tell us just a little bit about how you would gather some data, the first part, what you would do from specimens, and how old of things could you be successful?
Yeah, so they're too old for the technique that I was using, which is kind of an outdated DNA technique, just doing Sanger sequencing. And basically, you need large chunks of DNA for that. But if a specimen is really old, maybe over 10 years old, then the DNA starts to kind of fragment and break down. And so I can't use my technique for it. The chunks of DNA are too small. But there's all sorts of new DNA amplification techniques that have no problem with really small pieces of DNA. They're just more expensive.
Just out of curiosity, if I may, when you do your DNA studies or separations, I have a young friend working on, hang on here, a senior moment, phasmids, the walking sticks. And he grabbed a couple of legs on several of the specimens that I had. They were over 10 years old, if not a little bit longer than that, yet he was still able to get whatever information he needed. Are there different techniques that are used for pulling this information?
Well, I mean, 10 years is a rule of thumb. It all depends on how the specimen was cared for. If the specimen was immediately dried out and kept dry, it's probably good for longer than 10 years. If it was a moth that was maybe re-relaxed, like re-humidified, then the DNA will be damaged, and it probably won't be good, for over a year. Or for more
What
than a year.
damages the DNA? Are we talking about heat? We're talking about
Water.
I'm sorry?
Just water. And there are other things that will do it, like Vapona, which is a common fumigant or insect
Right.
deterrent. That'll break down DNA pretty quickly. But water is the big thing.
Thank you. Any more questions? With that, then I guess we would like to say thank you very much for a very interesting and great presentation.
Thank you.
When you get out this way, like I say, keep in touch with us. If you're interested, we'll put you on the mailing list and you can join us whenever you would like.
Yeah, absolutely.
All right, Kim, anybody else out there have any questions? All right, with that, I'm going to be signing